Good day, everyone. We are the reporters of Chapter 9 Gene Therapy. Here are the reporters Sanchez Alessandra, Santiago Edselin May, and Sumang Alaisa. Learning objectives. At the end of this unit, I am able to discuss the concept of gene therapy and its various forms, discuss and assess the benefits and detriments of gene therapy to human health, assess the issues, potential benefits, and detriments to global health. Let us now discuss what gene therapy is. Gene therapy is a technique that modifies a person's genes to treat or cure disease. It is a technique that uses genetic material, a piece of DNA, for the long-term treatment of genetic disorders. So, ang gene therapy ay isang medical approach kung saan pinapalitan yung defective gene sa cell ng isang tao para magamot o ma-prevent ang mga sakit. So, for example, gene therapy is like fixing a mistake in a computer program. Our bodies have tiny programs called genes, but sometimes there are errors. Gene therapy is like going in and correcting those errors to help our bodies work properly, just like fixing a computer bug where it helps it run smoothly. Approaches to gene therapy. Gene therapies can work by several mechanisms. First, replacing a disease-causing gene with a healthy copy of the gene. So, if there is a gene in our body that causes problem or disease, it's because it is not working properly. And yung gene therapy ay magagamit natin para mapalitan yung problematic gene with a healthy one. So, yung mga scientists ay mag-i-introduce ng panibagong kopya ng gene which is a healthy one na papalit sa faulty gene. Yung pamalit na gene na yon ang magte-take over ng role ng pinalitan nitong faulty gene na tutulong sa ating katawan na mag-function properly. At may potensyal na magamot kung ano man yung naging epekto ng faulty gene sa katawan natin. Next is inactivating a disease-causing gene that is not functioning properly. So, when a gene in our body is not working properly, gene therapy can act as a switch that can be used to turn off or inactivate the problematic gene. So, magagawa natin to by introducing a modified version of that gene that stops the gene from working. So, kapag na-inactivate natin yung disease-causing gene, mape-prevent natin ito na mag-produce ng harmful effects sa ating katawan. Lastly, introducing a new or modified gene into the body to help treat a disease. So, when a person has a disease because of a missing or malfunctioning gene, gene therapy can introduce a brand new or modified gene into the body. This added or modified gene is designed to perform a specific function that body's own genes can do properly or correctly. It is like giving the new body a new set of instructions to help it fight the disease or perform a task that it couldn't do before. This new or modified gene can help the body work better and fight off the disease, potentially improving the person's health. Next, we have the types of gene therapy. So, somatic gene therapy. In somatic gene therapy, the somatic cells of a patient are targeted for foreign gene transfer. In this case, the effects caused by the foreign gene is restricted to the individual patient only, and not inherited by the patient's offspring or later generations. So, dito sa somatic gene therapy, it involves modifying or correcting genes in the body's somatic cells, which are essentially all cells except the reproductive cells or the germ. So, ibig sabihin lang dito, yung naganap or yung pagbabagong ginawa sa somatic gene therapy, hindi siya maipapasa sa mga susunod na henerasyon because hindi nga naapektuhan yung patient's eggs or sperm. So, yung aim na tong somatic gene therapy ay para magamot or ma-alleviate yung specific diseases or conditions sa katawan ng isang tao. So, typically for therapeutic purposes without affecting their offspring's genetic makeup. Next, we have the germline gene therapy. In germline gene therapy, the functional genes, which are to be integrated into the genomes, are inserted in the germ cells. Example, sperms or eggs. Targeting of germ cells makes the therapy heritable. So, dito sa germline gene therapy, it involves modifying or correcting genes within the reproductive cells, which are the sperm or egg cells or embryos. So, kung sa somatic gene therapy, hindi siya naipapasa sa mga susunod na henerasyon or sa mga anak, dito sa germline gene therapy, changes made in germline gene therapy could be passed down to future generations because they affect the genetic material in the reproductive cells. So, yung aim nitong germline gene therapy ay para ma-prevent yung mga genetic disorders na pwedeng mapasa or mamana ng mga anak ng pasyente. So it potentially eliminates the transmission of certain genetic diseases to future generations. However, this technique is currently highly debated and ethically complex due to its long-term implications and potential unforeseen consequences.
Next, we have gene therapy strategies. Letter A, gene augmentation therapy or GAT. In GAT, simple addition of functional alleles is used to treat inherited disorders caused by genetic deficiency of a gene product. Example, GAT has been applied to autosomal recessive disorders. Dominantly, inherited disorders are much less amenable to GAT. Gene augmentation therapy involves introducing genetic material into a person's cell to replace or add a functional copy of a gene that's either missing or faulty. This aims to help the body produce the correct protein or function properly. In Figure 1, a gene therapy vector has been designed to treat the disease cells with a gene X. This vector was introduced inside the disease cells by various genes transfer methods. After a successful homologous recombination, the treated cells will show the presence of gene X product as well as normal. So, when a gene therapy vector delivers gene X into the disease cells and successfully integrates the gene using homologous recombination, it means the treated cells will start producing the product of gene X as well as functioning normally. This happens because the introduced gene X helps the cells create the necessary protein or function that was missing or faulty before the therapy. Letter B, targeted killing of specific cells. It involves utilizing genes encoding toxic compounds or suicide genes or prodrugs, reagents which confer sensitivity to subsequent treatment with a drug. To kill the transfected or transformed cells, this general approach is popular in cancer gene therapies. In targeted killing of specific cells means using a method that only affects and eliminates particular cells while leaving other cells unharmed. This could involve techniques or treatments that specifically target and destroy only certain types of cell, like cancer cells, without damaging healthy ones nearby. In Figure 2.a, direct killing of disease cells by two methods. The first method is the introduction of toxin gene into the disease cell, which when expresses toxin protein, the cells die. The second method involves incorporation of a certain gene, example TK, in the gene therapy vector which shows a suicidal property on introducing certain drug, example gencyclovir. In the first method, a toxin gene is inserted into the disease cell. When this gene is active and produces the toxin protein, it causes the cell to die. In Figure 3.b, assisted killing is another strategy of killing disease cells. Here, one method is to insert a well-known foreign antigen coding gene, which induces immune cells for the killing of the disease cells. Few more methods are based on immune cells activation in which a certain cytokine encoding gene incorporated into gene therapy vector and inserted into either disease cells or non-disease cells. This will lead to enhanced immune response followed by killing of disease cells. In the second method, a specific gene like TK is introduced into the disease cell using a gene therapy vector. This gene or TK has a special characteristic that makes the cell sensitive to a particular drug, such as gencyclovir. When the drug is administered, cells containing the TK gene become suicidal and die, targeting only those cells that have taken up the gene therapy vector. In this figure, in assisted killing strategies, one approach involves inserting a gene that codes for a recognized foreign antigen into the disease cells. This prompts the immune cells to recognize these cells as threats and triggers them to eliminate the disease cells. Additionally, there are methods that activate immune cells by incorporating a gene encoding specific cytokines into a gene therapy vector. When this vector is introduced into either disease or non-disease cells, it boosts the immune response, empowering immune cells to become more effective at targeting and killing the disease cells. This enhanced immune response helps in the elimination of the affected cells. Letter C. Targeted inhibition of gene expression. This is to block the expression of any disease gene or a new gene expressing a protein which is harmful for a cell. This is particularly suitable for treating infectious diseases and some cancers. Targeted inhibition of gene expression means using a method to specifically stop or reduce the activity of a particular gene. This could involve techniques that block the gene's instructions, preventing it from making its corresponding protein or functioning normally within the cell. In Figure 4, to inhibit the target gene expression in the cell, the antisense mRNA coding gene inserted vector or triplex forming oligonucleotides TFO or antisense oligonucleotide or ODN can be introduced, which will inhibit the gene expression either by forming DNA RNA triplex inside the nucleus or forming RNA duplex by forming complementary mRNA strand of disease protein coding mRNA. This may lead to blocking of disease causing protein expression. In this figure, to inhibit the expression of a target gene in disease cells, methods like using antisense mRNA coding genes in a vector or employing triplex forming oligonucleotides or TFOS or antisense oligonucleotides or ODNs are utilized. These approaches work by either forming a DNA RNA triplex within the cell nucleus or by creating an RNA duplex with the disease-causing mRNA, effectively blocking the production of disease-causing protein. This inhibition stops or reduces the expression of the protein responsible for the disease. In Figure 5, this is used for disease caused by mutation. The corrected gene will be swapped by the mutant gene X or M, then disease cells will become normal after the correction of mutation by gene therapy. In the last figure, in diseases caused by mutations, gene therapy can replace the faulty or mutant gene X or M with the correct version of the gene. By swapping the mutant gene with the corrected gene using gene therapy techniques, the disease cells can regain normal functionality. This correction helps the cells produce the right proteins or function properly, effectively reversing the effects of the mutation and restoring normalcy to the cells.
Next is methods of gene therapy. The goal of gene therapy research is to determine whether a new gene can be used to replace or inactivate a mutated gene to treat a disease or help the body fight a disease. For a gene to be delivered into a cell, a transporter is typically used. A transporter is known as a vector. A vector is made from an altered virus before the virus is used as a vector. Its viral genes are removed. Once the virus is modified, it is intended to transport the desired gene to the cell without causing disease. Once inside, the desired gene should restore the function of the protein. Vectors can be given intravenously, which means they are administered into a vein or injected into a specific tissue in the body. Others procedures, such as surgery, can also be used to deliver vectors into specific areas of the body. Sa madaling salita, ang gene therapy ay isang paraan kung saan ang mga bagong gene ay pinapasok sa katawan ng tao upang palitan o hindi gumana ang isang mutadong gene na nagdudulot ng sakit. Ang mga bagong gene ay dinala ng isang vector na karaniwang isang virus na binago para hindi na magdulot ng sakit.
Ang vector ay maaaring maipasok sa katawan sa pamamagitan ng intravenous na paraan o sa pamamagitan ng pag-inject sa isang tiyak na tissue o sa pamamagitan ng operasyon. Kapag nasa loob na ng cell ang bagong gene, ito ay maaaring makapagbalik ng normal na function ng protina. Ang vector ay parang isang transportasyon para sa mga gene. Ito ay karaniwang isang virus na binago ng mga siyentipiko para hindi na ito magdulot ng sakit. Ang trabaho ng vector ay dalhin ang mga bagong gene sa loob ng mga cell sa katawan. Sa madaling salita, ang vector ay parang isang delivery van na nagdadala ng bagong gene sa tamang destinasyon. There are mainly two approaches for the transfer of genes in gene therapy. A. Transfer of genes directly to cell inside the body in vivo. So, sa in vivo, ang unang paraan ay tinatawag na in vivo gene therapy. Ito ay nangangahulugan ang mga gene ay direktang ipinasok sa loob ng katawan ng pasyente. Halimbawa, ang mga gene ay maaaring ipasok sa pamamagitan ng pag-inject sa isang tiyak na bahagi ng katawan o sa pamamagitan ng intravenous na paraan, kung saan ito ay ipinasok sa pamamagitan ng isang vein. Sa paraang ito, ang mga gene ay diretso na pumapasok sa mga cell ng pasyente. Letter B. Transfer of genes into patient cell outside the body. Ex vivo gene therapy. Ang pangalawang paraan ay tinatawag na ex vivo gene therapy. Sa paraang ito, ang mga gene ay inilipat sa mga cell ng pasyente sa labas ng katawan. Una, kukunin yung mga cell mula sa pasyente. Pagkatapos, ang mga gene ay ipapasok sa mga cell na ito gamit ang mga vector. Kapag ang mga cell ay meron ng mga bagong gene, ito ay babalik sa katawan ng pasyente. Sa paraang ito, ang mga gene ay inilipat sa mga cell bago ito ibalik sa katawan. Ex vivo gene therapy. In this mode of gene therapy, genes are transferred to the cell grown in culture. So dito, yung mga gene ay tinatransfer into the cell grown in culture. Matapos ito, ang mga cell na binago ay pinipili, pinaparami, tsaka ini-introduce into the patient. Next is the use of autologous cells avoid immune system rejection of the introduced cells. Ang paggamit ng autologous cells ay pag-iwas na pagtanggi ng immune system sa mga pinapasok na cells sa loob ng katawan ng isang tao. Next is the cells are sourced initially from the patient to be treated and grown in culture before being reintroduced into the same individual. Next is, this approach can be applied to the tissues like hematopoietic cells and skin cells, which can be removed from the body, genetically corrected outside the body, and reintroduced into the patient body, where they become engrafted and survive for a long period of time. Ito ang paraan ng ex vivo gene therapy kung saan ang mga gene ay inililipat sa mga cell sa labas ng katawan bago ito ibalik sa pasyente. So, meron tayong 4 na uri ng cells na ginagamit sa ex vivo gene therapy. Una, yung hematopoietic cell. Pangalawa ay skin cell. Tas yung pangatlo, T cells. At yung pang-apat ay mesenchymal stem cell. In vivo method of gene transfer involves the transfer of cloned genes directly into the tissues of the patient. In vivo gene transfer, direkta nitong nililipat yung mga klonal mga tissue ng pasyente. Ginagawa ito sa mga kaso ng mga tissue such as brain cell that cannot be cultured in sufficient number outside the body. This is done in case of tissue whose individual cells cannot be cultured in vitro in sufficient numbers like brain cell and or where reimplantation of the culture cells in the patient is not efficient. Liposomes and certain viral vectors are employed for this purpose because of lack of any other mode of selection. In case of viral vectors, such type of culture cells were often used which have been infected with the recombinant retrovirus in vitro to produce modified viral vectors regularly. These culture cells will be called as vector-producing cells or BPCs. The BPC transfer the gene to surrounding disease cells. So, yung viral vectors ay ginagamit upang maipasa yung mga gene sa mga tissue ng pasyente. Yung karaniwang ginagamit ay yung culture na infect ng recombinant retrovirus sa labas ng katawan upang makapag-produce ng modified viral vectors. Tinatawag ang mga cell na ito na vector-producing cells or yung BPC. Ang mga BPC ang naglilipat ng mga gene sa mga kalapit na sakit ng mga cell. The efficiency of gene transfer and expression determines the success of this approach because of the lack of any way for selection and amplification of cells which take up and express the foreign gene. The efficiency of gene transfer ay nakasalalay sa epektibong paglipat at pagpapahayag ng mga gene. Ito ay dahil walang ibang paraan para sa pagpili at pagpaparami ng mga cell which take up and express the foreign gene. Sa madaling salita, yung in vivo gene therapy transfer involves directly transferring clone genes into the patient tissues, particularly when other methods of cell culture or reimplantation are not feasible. Liposome and viral vectors are employed due to the lack of alternative selection methods. In this figure, therapeutic genes have to be delivered to specific target sites for a specific type of disease. This table describes the list of such disease and their target site for gene therapy. Advantages and disadvantages of gene therapy. Advantages of using gene therapy. Gene silencing. In the case of an HIV-infected person, gene therapy and gene silencing can protect the patient from pain and suffering before the disease progresses. Gene therapy is potentially used to eliminate and prevent hereditary diseases such as cystic fibrosis. It is also a potential way to treat heart disease, AIDS, and cancer. Disadvantages of gene therapy. The novelty of gene therapy method is one disadvantage. Stimulation of immune response. The gene injected by a virus may cause immune responses due to the presence of the virus inside the body and the pathogenic potential of viral vector. In one case, the viral vector could improve its ability to cause illness. Generation of genetic disorder due to the presence of multigene. The genetic material transfer may not necessarily enter the target cell. Even if it does, it may not be placed at an appropriate place in the genome.
That's the end of the chapter 9. Thank you for listening and have a good day.